Chapter 7, Section 16, Post-Translational Regulation. Here we're going to be looking at regulation of enzymes, not through allosteric inhibition like the post-translational, the feedback inhibition from Section 15, or the transcription translation regulations from Sections 13 and 14. Here we're going to be looking at, okay, you have a protein that's made, and you're either going to activate it or inactivate it by changing its shape. Changing its shape not by allosteric means, but by a covalent modification. You are going to bind something to it. You're going to bind something to it, a phosphate group, a methyl group, or something like that, because by adding a inorganic phosphate group or a methyl group, you are changing secondary structure, change tertiary, change quaternary, change shape, give it function. Remove of the phosphate group, the methyl group, reverts it back to its original shape, loss of function. And yes, you can also look at it by adding the addition of the phosphate group, changes, the free energy, the delta G and all that, which, imbues, exergonic energy into an endergonic reaction, or whatever, how you want to think about it. We're going to see that the common modifiers that we can see that are going to be added to certain proteins to activate or inactivate them. Adenosine monophosphate, adenosine diphosphate, adenosine triphosphate, uridine monophosphate, AMP, ADP, UMP, these are going to be added directly to, whereas the ATP, it's going to be having the inorganic phosphate group transferred from. The P2 signal transducing proteins are a really good example of this. We're going to find these bacteria, archaea, some plant plastids. So not all plants, just this plastids, a small subgroup of plants. We see that these P2 signals are highly involved with nitrogen metabolism. They regulate this. They specifically regulate the transcription factors, the enzymes, and any membrane transport proteins. The addition of these groups to these proteins either activates them or inactivates them. The modifications that are mostly going to be seen here, the addition of UMP, AMP, uridenylation, adenylation, and phosphorylation, just the transfer of the terminal phosphate from ATP to the protein itself. This is going to be thought of as post-translational modification. Okay, the protein's made, the protein's just sitting there in its inactive form. You add some of these, you activate. Some of these proteins are made, they're already active, and it's the addition of a UMP group to it is going to interfere with its activity. It's going to change its shape, thus making it lose its function. You look here, figure 7.35 from the textbook. High to low levels of glutathione. Low levels, low concentration levels of glutathione. GLND, not bound, because there's too little glutathione in the cell cytoplasm. So it's going to take P2, and it's going to add UMP, uridyl monophosphate, to it. This UMP going to interact with this GS-AMP, giving us glutathione synthetase. UMP, AMP interact. It's going to remove the AMP group, which is inactivating GS right now, to make it active. And, glutathione synthetase makes more and more glutathione, which means you're now increasing in glutathione concentrations. You get to high enough levels of glutathione concentration. Glutamine, I keep saying glutathione is glutamine concentrations here, not glutathione, glutamine. Glutamine levels get high enough. What's going to happen now is that glutamine is going to feedback inhibit GLND. GLND, when it's bound to glutamine, is now going to do the reverse of what you see here. It's going to take PI-UMP, turn it back into PI. PI takes glutathione synthetase and adds AMP to it. So you're now seeing the reverse of the reactions due to low or high concentrations of glutamine. Now, it's not a one and done with GS. GS is going to be a cluster of polypeptides. And you see as the glutamine concentrations increase, more and more of the polypeptides, proteins in this complex, will get AMP covalently added to them. As more and more get added, the activity of this complex will decrease. It goes from fully active, partial active, to completely inactive, depending upon how many of polypeptides, the proteins in that complex, have been, adenylated. Here's everything I just said. GLND senses glutamine, okay, interacts with glutamine. It's not a feedback inhibitor, but it the feedback changes its activity. Too low signals that we need to have ammonia simulated. GLND is not bound to glutamine. It takes UMP to P2, forming P2-UMP. P2-UMP targets glutamine synthetase, adenyl transferase. Stimulates the release of glutamine synthetase, which means more and more glutamine will be made. More and more glutamine gets made. GLND does the reverse. Takes the UMP off of P2. P2 then takes GS, adds, AMP back to it, inactivating it. Another post-translational regulation mechanism we see in prokaryotes is those sigma factors. Remember, we talked sigma factors early on when we were talking stress response proteins. These are going to be proteins that control metabolic pathways and control expression of different regulons. These anti-sigma factors, which can then inactivate sigma factors. Sigma factors control the regulons, the metabolic pathways. The anti-sigma factors controls the sigma factors. RPOE and RSEA are two great examples. Under normal conditions, RPOE is tethered to RSEA. How it's tethered, don't know, don't care, don't worry about. They're just held. RPOE is just held here at RSEA, which is a cytoplasmic integral membrane protein. Okay, so it's held here, sequestered away, until there's some form of damage to the membranes. Damage to the membranes, well, damages RSEA, which releases RPOE. RPOE is a transcription activator. It goes and binds to certain promoter regions, and then the next thing you know, you get certain genes being transcribed, envelope repaired, DNA-K. DNA-K goes through and will turn over proteins, recycle the polypeptides, get the amino acids back. These genes for repair are going to go through, replenish, replace any damaged phospholipids, lipopolysaccharides, things like that. Under quote-unquote normal, everything's sequestered. Anti-sigma factor RSEA is holding the sigma factor RPOE. Damaged, anti-sigma factor goes away. Sigma factor RPOE is released, and notice it goes to multiple places on the chromosome. Different operons, different genes, different operons at different places. Remember, regulons, and will initiate transcription of these genes under this membrane damage stress event.